All right, we're going to start on the immune system. We have 2 lectures to get through chapter 21. So obviously the immune system is going to be a very complex system that you guys need to know. For this exam, you guys are going to have some questions on chapter 20, but they're going to be minimal in comparison to chapter 21, because chapter 20 is just a lead-in to chapter 21, where chapter 20 is just telling you all of the organs that are going to be responsible for your immune system, and all of the organs that are going to be working with your immune system. Chapter 21 goes over the actual immune system and what it is doing in the body. So this is the hard-hitting physiological information that you need to know for the lecture exam. There are 2 chapters on, or not 2 chapters, there are 2 lecture PowerPoints on this because it's a lot of information, okay?
And this will be on an exam,
not tomorrow's
tomorrow,
exam.
not the next one, but the one after that? Or it will be on the next one?
It'll be on exam 3. So
Okay.
exam 2 is tomorrow, and then this is the exam 3 information.
Okay.
So we will most likely be through all of the information we need for exam 3 next week, because your exam on all of this information is going to be the week after that. Okay. All right, so this information should be, like, you guys should be excited to learn about this chapter. It's a lot of information, but it's also really, really interesting to know what's happening with your immune system, right? Like, every single person in this classroom has been sick at some point in time, and it's kind of nice to know what cells in your body are doing what. So then you can be the obnoxious person in the room when you get sick or when somebody's sick and be like, okay, so what's happening is you have these cells, and these cells are working with these cells, and you can just be the annoying know-it-all in the classroom. It's a lot of fun. And then you can also tell them what cells aren't working as well, if they're not getting better faster, or things that they should take that could possibly help their immune system and its function, things like that. Okay. So we're going to go over the 2 types of intrinsic systems for the immune system. You have innate and an innate defense system. This is a non-specific defense system for the immune system. And then you have the adaptive defense system, which is specific. So the innate is going to be your first and second lines of defense for your immune system. The first line of defense that it forms is going to be your external body membranes, basically your barrier between you and the outside world. So your skin, any of your like membrane surfaces, the mucosa, so all of the mucus forming surfaces of your body. And then the second line of defense is going to be all of the things that lie directly underneath it that are going to be your first response to an invasion. And that's going to be things like antimicrobial proteins, phagocytes, and other cells that are going to inhibit invaders from getting into your body. Okay? For your adaptive defense system, this is the third line of defense. This is going to attack very particular foreign substances. This is your like long-term immunity that's really going to keep you from having continuous attacks from the same foreign invaders. And this is also going to be something that responds to cancer, and it's going to have a slower activation rate, but it's going to have a longer period of time that it's active and actually killing and fixing things in your body. The immune system is a functional system rather than an actual organ system. So this utilizes different organ systems that have to work together to do this whole immune function. Your inner and adaptive defenses are going to be intertwined with each other. They don't work separate from each other. They're going to work together to fully kill off any invasions that happen in your body. So both of them can release and recognize many of the same defensive molecules. Innate defenses will have specific pathways for certain substances, and the innate responses will release proteins that alert cells that the adaptive or cells of the adaptive system to the foreign molecules. So your innate defenses are going to communicate directly with your adaptive defenses so that they can activate them, which is also another reason why your adaptive defenses are a little bit slower to activate, because they require a lot of signals to occur, and a lot of those signals are going to come from that innate response. All right, so this is a very good flow chart for looking at the overview of innate defenses versus adaptive defenses. For innate defenses, you have your first line of defense are your surface barriers, the skin and the mucous membranes. Your second line of defense is the internal defenses. Phagocytes, natural killer cells, inflammation, antimicrobial proteins, and fever are all going to be part of this second line of defense. For your adaptive defenses, you have two types. You have humoral immunity and cellular immunity. Humoral immunity and cellular immunity are both going to be moderated by your lymphocytes. Lymphocytes are how common for your white blood cells? Number what? 2. So lymphocytes are number 2 for most common white blood cells in your body. And that makes sense considering that they make up all of your adaptive defenses. They have to be fairly numerous in your body. What do neutrophils do? Do you guys remember? Which you guys have to know for tomorrow. They attack what? Bacteria and other things. So neutrophils do a lot of phagocytic activity. So because neutrophils are very phagocytic, they're very numerous because they'll attack pretty much anything that shouldn't be in your blood, right? So neutrophils are the most common, and then they are part of your internal defenses, and then your lymphocytes are the second most common, and they make up your adaptive defenses. So for your B cells and your T cells, remember that lymphocytes, there are two types. You have T lymphocytes and B lymphocytes. We also call them T cells and B cells. You should automatically know when you see T cells and B cells that they are lymphocytes. Okay? The innate system uses the first and or second lines of defense to stop attacks by pathogens. A pathogen is any disease-causing microorganism that enters the body. So for the surface areas, you have skin and mucous membranes and their secretions. So their secretions are very important because the secretion has enzymes and bacteria and toxins that it can get rid of bacteria and toxins. So it's going to have enzymes that can break down the bacteria. It's going to have usually some weak acidity to it, and all of this is going to be important for what your epidermis is primarily made of, which your epidermis is primarily made of what protein? Keratin. So your epidermis is primarily made up of layers of keratin, right? And keratin is very, very important to have in that outer surface because it's resistant to weak acids and bases and the bacterial enzymes and the toxins that can get onto your skin. So you, despite what all of the commercials say about, like, you know, putting some sticker thing on your foot and it pulls the toxins from your body or something like that, you don't have toxins on your skin because your skin is resistant to toxins. If the toxins could pass from inside of your body to the outside, then they could also pass from the outside to the inside. And your epidermis prevents that because it is made up of keratin, which is resistant to these toxins. Basically, all you're doing when you put that sticker on your foot is you have a color change because it is made up of a certain type of acidity, and when that acidity touches your foot, it causes like a pH change, and it causes the strip to change colors. It makes people feel like they're cleaner when they're done, not actually doing anything. The mucosa are very important because they are going to provide mechanical barriers, and then your skin also provides a physical barrier to most microorganisms. They're not going to be able to penetrate the barrier of your skin. They are both going to be able to produce protective chemicals that will inhibit or destroy microorganisms. So you need to know about these two things, the acids and the enzymes. For the acids, the acidity of the skin and some mucus secretions will inhibit growth of bacteria. And we call this acid that is regularly around the skin and the mucous membranes the acid mantle. Okay? You may have heard of the acid mantle before. what is found in very, very large quantities in your body? On your body and in your body?
Bacteria.
Bacteria. How many of the cells found for your body are your own? Are produced by your body, contain your DNA, and are yours?
Less than the bacteria, right?
Far less. You guys remember from 160?
Yeah.
Has everybody taken 160? We tell you in 160. Maybe? Does anybody want to take a guess? How many of the cells of your body do you think are your own?
That was like 10 or 20 or something. So small.
Yeah. Which one of those?
10.
10. 1/10 of the cells that are found for your body are your own. The rest of them are bacteria and other microorganisms. Which cells are bigger, eukaryotes or prokaryote? How did I word the question? Which one?
You or
Yeah, which one is bigger?
You.
Eukaryotes, right? What are we made up of? Which one do we fall into? Eukaryotes, right? So our cells are very large compared to prokaryotic cells. Where does bacteria fall? Prokaryotes. So bacteria are much smaller cells. They're much more numerous on your body surfaces and inside of your body. You have bacteria that lines your digestive tract. You have bacteria that's found in your mouth. You have bacteria that's found all over your body. And those microorganisms are going to account for many, many, many more cells than the cells of your own body. So only about 1/10 of the cells of your body are your own. Okay? So it's very important that because only 1/10 of your cells are your own, on these body surfaces you want to keep those cells from over multiplying. If they over multiply and they get very, very abundant, what kind of disorders can you have happen on the skin? So, yes, a little bit. So acne can be from overgrowth of bacteria. Do you guys remember how a pimple forms? What causes it?
Inflamed sebaceous gland.
It's basically an inflammation in the sebaceous gland because it got backed up or it got a bacterial infection. So high amounts of bacteria on the skin because of incorrect acid mantle development could be a cause of acne. There are a lot of different treatments for acne. It's why some people say like, oh, Proactiv works for me, and some people say some other treatment works for them because they have different causes for their acne. So their skin condition is going to respond to a different treatment. Each of those does like a different thing for changing the acid mantle and how it cleans the pores and everything else. Anything else? You guys can think of. Is bacteria the only microorganism found on your skin? What is a common condition that women can get?
Oh, yeast infection.
Yeast infection. So what are yeast? Microorganisms. Where is yeast found? Are you bread? Yes, we put yeast in bread because of its activity with sugar. Where is yeast found? Typically in moist areas. So what would be some moist areas of the body? Crevices like what? What?
Privates. Genitals.
What's the anatomical term for those areas that yeast would grow? The vagina is an internal organ. If you have yeast in the vagina, it's a yeast infection. which we'll talk about when we get to reproductive, but when people are like, oh, my vagina hurts, they're not talking about their vagina, they're talking about their vulva. You know, the external features are like the vulva. You could say mons pubis, maybe. That's where the hair grows. And mons pubis sounds more badass than vagina, but everybody likes the word vagina for some reason because they think it's dirty. I don't know.
So the yeast grows on like epithelial tissue?
So yeast is found on the outside of the body, but we're talking about what you would call the region that you would find yeast that is a crevice for the private area, as somebody
Groin.
said, or where else you would find them. Groin is not the correct. Starts with an I. Inguinal.
All right.
So you have the inguinal regions where you might have higher amounts of yeast. Where else might you find yeast? Where else do people have a high amount of moisture that's kind of like a crevice?
Armpits.
What do we call that region? Anatomical terms only. Axillary. Okay, so yeast grows very commonly in the axillary regions and in the inguinal regions, but you can find it anywhere basically that the body makes a crevice. Yeast is found all over the body, but it's found in higher quantities in areas where you have crevices, where sweat collects and you have higher moisture. If the acid mantle is not correct and it doesn't inhibit the growth of yeast, you can get yeast infections. Yeast infections can occur anywhere on the body, and you see them very commonly in patients that have that are morbidly obese because they have rolls of skin that create crevices throughout the body, and they'll end up with yeast and bacterial infections. Because also with a larger surface area with more skin to cover, the acid mantle is not going to be able to cover all of that surface area. It's not made to just extend with tissues extending as well. So you'll have a higher incidence of bacterial and yeast growth for patients that are morbidly obese. You also have enzymes that are found for this first layer of defense. So you have lysosome of your saliva, you have lysosomes in the respiratory mucus, and you have lysosomes in the lacrimal fluid, and these will kill a lot of microorganisms. It's one of the reasons that they say that tears are like healing tears, right? Because tears actually do contain a lot of antibacterial properties and enzymes that will kill off a lot of things. So when something gets into your eye and your eye tears up, it's very good for your eye to do that because it's going to clear out all of the debris and attack any foreign objects that happen to get into your eye. You also have enzymes in the stomach that will kill pretty much any microorganism, and also the high acidity of the stomach is going to help a lot with killing microorganisms. Mucin is a sticky mucus, and it lines the digestive and respiratory tracts so that it can catch and trap microorganisms. So mucin, you need to remember what mucin is made from. What makes mucin?
Mucus.
No.
Mucus.
Trick question. What is mucin? What is it made of?
Mucin.
No. What is mucin?
Glycoprotein.
It is a protein. So mucin is a protein, and mucin is a protein when it mixes with water, it becomes very sticky, and it basically dissolves in the water so that the water will become mucus. Mucus is made from mucin dissolving in water. Okay? That's an important thing to know. You need to know how mucus is made. Mucin dissolves in water, that makes mucus. You're going to learn more about defensins as we go along, but defensins are antimicrobial peptides, and they inhibit microbial growth because of how these proteins work. You have other chemicals like lipids that are found in sebum. Do you guys remember what sebum is? Piece of paper. Name and date. Tell me what makes sebum. everybody has turned in.
Christy's like, full time. Really? You guys messed it up, or yes? What should I do? I was reading it and I use a chopstick to hold it. There was like, I'd read like a paragraph and then press
Alright, so what is sebum?
Oily.
The oil. So what makes oil? What makes sebum? Sebaceous oil glands. Okay? So you have lipids in sebum and dermacidin in sweat, and these can be toxic to some bacteria. So just the oil that you make on your skin can kill some bacteria. And then you have dermacidin, which is going to be found on the dermis and kills bacteria. The respiratory system also has some modifications that will stop pathogens. You have mucus-coated hairs in the nose that trap your inhaled particles. And then you have the cilia of the upper respiratory tract that will sweep dust and bacteria-laden mucus towards the mouth. Surface barriers that are breached by nicks or cuts will trigger the internal second line of defense. Okay? So all of this first line of defense is just when your system is normal. When you have no cuts, you have no openings to the inside of the body that should not be there. Okay? So the first lines of defense are just going to be normal function for your body to prevent things. When the barrier is breached, then you are going to have the second line of defense activated in your deeper tissues. So again, these tables are very helpful because it takes you through all of the different parts of each system and what they do to help with your immune system, so their protective mechanisms. So these tables are important to know. You want to know this because it is helpful. I didn't go over every single thing on here, but you want to know all of their protective properties. So you have like gastric juice and urine are on here, the acid mantle of the skin, keratin, nasal hairs, cilia, and then you also have an acid mantle for the vagina, for the female reproductive tract. And the acid mantle in the vagina is going to protect the female's reproductive system by killing off most bacteria and fungi, which is why when yeast overgrows, then it's called a yeast infection. And you don't want an infection to go into the female reproductive tract because it can find its way into the uterus. And when an infection finds its way into the uterus, we call it pyometra. And for people, you can treat it with like flushing and heavy antibiotic drips because we have a larger uterus that is more open and it's easier to get the infection out. But for other mammals like dogs and cats, it means we have to remove their uterus because they have a horned uterus and the infection can get trapped in the horns and they can die from it if you don't remove it. For humans, we don't have to do a hysterectomy if you get an infection. It's just a more intense treatment because you have a deep internal organ that gets an infection. So the vagina helps prevent that a lot because the vagina has an acid mantle to prevent those kinds of infections from getting in. All right, so now we're going to go into the second line of defense. This is going to involve a lot of cells and chemicals. So this is where you start to get into like more heavy information that is not quite as intuitive, and you guys have some new information here. So you have phagocytes, you have natural killer cells, which everybody thinks natural killer cells is something that like I made up, and I didn't. It's actually a thing in your body. We have cells that we call natural killer cells. You have your inflammatory response. Your inflammatory response is going to involve macrophages, mast cells, your white blood cells, and inflammatory chemicals. Which of your white blood cells is going to be responsible for activating for inflammatory response? Got a note for tomorrow. You have 5 choices. Basophils. So basophils are going to help with the inflammatory response of your white blood cells. Remember that for all 5 of your white blood cells, you have to know the functions, and you have to know them for tomorrow. You also have the antimicrobial proteins. Your antimicrobial proteins are going to be interferons and complement proteins. We're going to go over complement proteins and how complement proteins form. It's a very cool process. It's basically, they're called complement proteins because they build onto each other in a complementary fashion, and they form a pore that basically just lets all of the inside contents of the cell out. So complement proteins are a really cool defense mechanism that will go onto a cell that you need to kill and just form all these holes in the membrane that aren't supposed to be there to lyse the cell. And then you have fever for the second line of defense as well. So many second line cells have pattern recognition receptors, and these pattern recognition receptors are going to recognize and bind tightly to structures on microbes. And when they do this, it will disarm the microbe before it can do harm in the body. For your phagocytes, the white blood cells that ingest and digest foreign invaders are going to make up some of your phagocytes. Which other white blood cell is there other than the one that's listed on there that's going to be phagocytic? Monocytes. So monocytes, as they move to peripheral tissue, how do they move to peripheral tissue? What do we call that process? It uses chemicals to get the monocytes to migrate out of the blood into the tissue. You guys are scaring me. It's all information you have to know for tomorrow.
Still got 24 hours.
Hemo something. No. No. Chemo.
Chemotaxis.
Chemotaxis. So chemotaxis is going to be how monocytes move to the peripheral tissues, right? So your neutrophils are your most abundant phagocytes, but your neutrophils will die when they're fighting a microorganism, so they don't really have another system where they are going to just keep activating more of your defense system. They're just going to die when they are exposed to infectious material and try to kill that infectious material. Your macrophages, these develop from monocytes, and these are your chief phagocytic cell. This is the most robust phagocytic cell because you have a lot of different types. You have free macrophages and you have fixed macrophages. For free macrophages, they're going to wander through tissue spaces. So for example, you have alveolar macrophages that kind of wander between all the alveolar connective tissue and wait for like something to possibly break the barrier inside of your lungs, and then these free macrophages are going to attack whatever broke through. Fixed macrophages are permanent residents in some of your major organs. So where they are going to be needed the most, you're going to have these fixed macrophages that are going to monitor exchange of blood material with the tissue, and they're going to kill anything that should not be going through. You have stellate macrophages that are found in the liver, and then microglia, which are found in the brain. And we kind of went over the microglia in the brain before, right? Okay. And then the stellate macrophages in the liver, we'll go over more of their function when we go over the digestive system. So phagocytosis, you need to know this whole process for phagocytosis. Each of these steps is very important to know. Phagocytosis starts when a phagocyte recognizes and adheres to a pathogen's carbohydrate signature. So remember that you have on all of your cells of your body, you have a carbohydrate signature that is made up of all of these sugar chains that extend out from the cell into the extracellular space, right? You guys remember this vaguely from 241? What do we call that sugar signature on the cell? It's made up of glycoproteins and glycolipids, a lot of transmembrane proteins and lipids that have a sugar chain that comes off of them that extends out into the extracellular fluid. Huh?
Extracellular matrix?
Mm-mm. Starts with a G. It's a real weird word. When you took 241 with me, I introduced you guys to the word because I like it and you have to know it for the immune system.
Does it start with glyco?
Mm-hmm. So much nervousness. The awkward silences, I'm told, are better for you guys. I'm supposed to let you guys think. Everybody's like, we have an exam tomorrow. I'm not doing this. Glycocalyx. So the glycocalyx is the sugar signature for all of your cells. So the way that your immune system is going to recognize that something belongs to your body or not is because it's going to recognize the glycocalyx. If the glycocalyx is not one that it is programmed to recognize, that means it's probably not supposed to be in your body and it's going to attack it. Okay, so some microorganisms have external capsules that can hide their surface carbohydrates. It can hide their glycocalyx. And this helps them evade your monocytes so that they don't get phagocytized. But we have a way to basically go around this, which the way to get around the capsule is to do opsonization. So the immune system has antibodies and complement proteins that can be used as opsonins, and those will coat the pathogen and basically start the process of breaking it down without needing phagocytosis. And then once those bind, the phagocytes will recognize these opsonins, they'll recognize the antibodies and the complement proteins, and they're like, hey, what are you guys doing? Let me help you with that, and then they can phagocytize something even if they don't recognize it as being foreign. Okay? So your antibodies and complement proteins can act as like little handles for the phagocytes to find these hidden microbes and kill them. Alright, so for phagocytosis, you want to know the steps that are going to be shown all together in this. I think this is just one random weird image. Yeah. That's just one random weird image to start. I don't know why. I always forget to take it out, but that was a random weird image. Just ignore that one. So you need to know the steps of phagocytosis. Oh, I think it's because that's showing one. I just forgot to delete it. So it usually does like one step, and then it shows one step on the diagram, and then it shows the second step, and then it shows the second step on the diagram, and that's just obnoxious and it takes up extra slides, and I just forgot to delete the first one. So for the second step, you have cytoplasmic extensions or pseudopods that are going to bind to engulf a particle in the vesicle that is called the phagosome. The phagosome then fuses with a lysosome, and that forms your phagolysosome. The phagolysosome will acidify, and the lysosome the lysosomal enzymes will digest particles of the ingested microbe. And then the ingestible and residual waste is exocytosed from the phagocyte to be removed from the body. So this is the image you want to pay attention to because it has all 5 steps put together. So step 1, the phagocyte adheres to the pathogen or debris. It has to recognize it as foreign and then bind to it. During this step, you are going to extend pseudopods. What does pseudopod mean? Fake foot. Pseudopods are the fake feet of your phagocytes that move in an amoeboid-like fashion. And so you have it pushes cytoplasm against the membrane to make it extend out. And then it's going to extend out as two little fake feet around the microorganism. In step 2, those feet are going to come together and it forms a vesicle around the microorganism that can then go into the cell. Step 3, that phagosome is going to form with a lysosome to form a phagolysosome. And then that phagolysosome, you're going to have the lysosomal enzymes destroy the pathogen and break it down. Then the 5th step is to get rid of all of that broken down material as waste from the cell through exocytosis. Okay? Is everybody okay with these steps? You have to know these steps. It is important. So some pathogens are not going to be killed with the acidified lysosomal enzymes. And this process is where you are going to trigger helper T cells to produce a respiratory burst, which will kill the pathogens that are resistant to lysosomal enzymes. It does this by releasing cell-killing free radicals, producing some oxidizing chemicals, and then increasing the pH and osmolarity of the phagolysosome. So when you have something like tuberculosis bacteria, you're going to have these helper T cells that trigger the macrophage that has the bacteria that it can't quite break down. It's going to trigger it to produce a respiratory burst, which will kill the pathogens that are resistant to them. And it does this with these 3 steps. Okay? And then you have defensives in the neutrophils will also help with this by piercing the membrane of the pathogen so that it makes it a little bit more I'm using the wrong word. It makes it more permeable to the enzymes, so the enzymes can go into the microbe and break it down. So if the enzymes are not reacting well, the defensins from neutrophils piercing the membrane of the pathogen before it gets introduced to these lysosomal enzymes will help with the breakdown process. Does that make sense? Alright, so non-phagocytic large granular lymphocytes that police the blood and lymph we refer to as natural killer cells. We call them natural killer cells because they can kill cancer and virus-infected cells before the adaptive immune system is activated. They attack any cells that lack your cell surface receptors, so any cells in your body that don't have your glycocalyx, the natural killer cells will go to kill them. The natural killer cells work by inducing apoptosis. So they signal for cancer cells and virus-infected cells to undergo apoptosis. What is apoptosis?
Programmed cell death.
Programmed cell death. So it's going to tell them to undergo cell death, go kill yourself, we don't need you here. So that's why they're the natural killer cells. They're just going to go naturally tell these other cells, go kill yourselves, and then they're going to implode, basically. Okay? So they secrete some potent chemicals that will also enhance the inflammatory response. So when they find these cells and they find that they're not right and they need to undergo apoptosis, another thing you'll need is as these cells break down, as they implode on themselves, they're going to produce a lot of waste material that needs to be broken down further, and it needs to go into the lymphatic system to be fully processed. So in order to do that, one of the biggest responses that you have for your body to flush things into the lymph system and into the lymph is to have inflammation. Inflammation will cause a huge flood of fluid from the interstitial spaces into that area, and it just washes everything into the lymphatic system. So having that inflammatory response from these natural killer cells will help in removing the debris that it creates by telling these cells to kill themselves. Inflammation is triggered whenever the body tissues are injured. Injuries can be because of trauma, heat, irritating chemicals, or infections by microorganisms. And you have a lot of benefits to inflammation. It prevents the spread of damaging agents, it disposes of cell debris and pathogens, and it alerts the adaptive immune system to an infection. And it's also going to set the stage for repair. Having this flood of tissue come in and removing all of the bad things is going to allow all of your cells that remain to undergo repair and to replace themselves at a steady rate without being attacked by all of these microorganisms. There are 4 cardinal signs of acute inflammation. So those are redness, heat, swelling, and pain. Sometimes there's a 5th sign, which is impairment of function, but it's only seen if movement or use of the area is hampered, and usually that 5th sign is when you have excessive swelling that is not going down and the inflammation is acting abnormally. For the stages of inflammation, you have inflammatory chemical release. So histamine is your primary inflammatory chemical. It is primarily going to be released by mast cells. That is the histamine is your key inflammatory chemical, primarily released by mast cells. What else releases histamine? What other cell? Basophils. So basophils will also release histamine. And histamine is going to cause vasodilation and increased vascular permeability. So increased vascular permeability means that the endothelial cells become more permeable. They're a little bit easier for cells to squeeze through. What kind of cells are going to be squeezing through the endothelial cells in your capillaries? Which white blood cells? Talked about this process earlier and what it's called. Monocytes are primarily going to utilize this increased vascular permeability because they undergo chemotaxis. Okay? And that's going to be your phagocyte mobilization, is vasodilation and increased vascular permeability, and then you increase phagocyte mobilization, which is going to increase their response to chemotaxis, and they are going to migrate out into the tissues to attack any foreign invaders. So vasodilation causes hyperemia, which is congestion with blood, and that leads to the redness and the heat that you get with inflammation. The increased capillary permeability causes exudate. Exudate is fluid that contains a lot of clotting factors and antibodies that leaks into the tissues. And this causes the local swelling or the local edema. The swelling also pushes on nerve endings, and that is going to result in your pain. So this is where you get all of the creation of the effects of inflammation that somebody might feel. For phagocyte mobilization, you guys need to know these steps. You have leukocytosis is the first step. This is release of neutrophils from the bone marrow in response to the leukocytosis inducing factor from the injured cells. Margination is when the endothelial cells of the capillaries in that inflamed area will project cell adhesion molecules, which are called CAMs, into the vessel lumen. The CAMs are going to grab onto passing neutrophils, and that causes them to slow. And when they slow down, they end up rolling along the blood vessel. And when they roll along the blood vessel, they are going to be able to cross through those endothelial cells. So as they roll along, those neutrophils undergo diapedesis. Diapedesis is when the neutrophils flatten and then start to squeeze between the endothelial cells, moving into the interstitial spaces. And then they're going to do chemotaxis, which the inflammatory chemicals will act as chemotactic agents, where these inflammatory chemicals signal for where the cells are supposed to go, where they're supposed to migrate to. That chemotatic property of the inflammatory chemicals is going to promote positive chemotaxis of the neutrophils towards wherever the injured area is. So basically, as chemotatic agents, what the neutrophils do is they are going towards areas where you have high amounts of this chemotactic agent, and it's just going to move towards those higher concentrations because that indicates where the injured area is. So it does chemotaxis by basically following a scent trail of these chemotatic agents to where it has the highest density, right? It's like if you smell cookies baking and you want to find out where the cookies are being baked, you follow the smell until you get to wherever those delicious, amazing cookies are being baked, right? Same thing with pie. When you walk into the house, everybody knows where the pie is right away, which everybody's going to have when we get to Thanksgiving, I'm sure, is figuring out where the pie is first when you walk in the door. So as the attack continues on the body, monocytes will arrive a little bit later. 12 hours after leaving the bloodstream, they're going to be transformed into macrophages. And these late arrivers will replace the dying neutrophils, and they remain in the tissues later for continued cleanup while the body is undergoing repair. Okay, so this shows you the events of phagocyte mobilization. So you start with leukocytosis. The neutrophils will enter the blood from the bone marrow. Then you have margination. The neutrophils will start to cling to the capillary walls. Diapedesis, the neutrophils will flatten and squeeze out of the capillaries, and then they undergo chemotaxis, where the neutrophils will follow the chemical trail of these inflammatory agents to the site of injury and start to destroy the microbes. As these neutrophils die, you need a more long-term response. So about 12 hours after the injury occurs, you'll have monocytes that are becoming phagocytic that are going to stay around for continued cleanup and to get rid of the debris left behind by these dying neutrophils. This is just a table that takes you through the responses in the body to tissue injury. You don't have to memorize this. This is just a helpful study tool to go over what we talked about. Okay? All right, pus is a creamy yellow mixture of dead neutrophils, tissues, and cells, and living and dead pathogens. An abscess occurs when collagen fibers are laid down, walling off a sac of pus, and a abscess sometimes needs to be surgically drained or surgically removed from the body. Usually what we do with an abscess is you just have to lance it because you have to pierce that collagen coating around it to expel the pus from this little sac. So the primary way to get rid of an abscess is to lance it, and you lance it using some kind of a small piercing tool. A lot of people, if they develop an abscess, will even just take care of it at home because we live in America and nobody likes to use our healthcare system because it's expensive and it's terrible and nobody likes to use it. So you just get like a needle and you heat it up until it's sterilized, and then you just pierce the skin and squeeze out all the pus. Very easy to clean, and then you clean it out with antiseptic. What antiseptic is good to use for the body? Betadine or iodine. Which one should you not use?
Hydrogen peroxide.
Hydrogen peroxide is very, very bad. Okay, so some bacteria, like tuberculosis bacilli, they resist digestion by macrophages and they can remain alive inside the body. They are basically going to form like tumor-like growths where they form these little pockets of this bacteria that can't be broken down. And these little tumor-like growths or pockets are called granulomas. And these are areas of infected macrophages that are surrounded by uninfected macrophages and an outer capsule. The bacteria can remain inactive forever, or if the person's immunity decreases, they can break free and become activated, causing the disease of tuberculosis. There's also, there's like a picture circulating around on Facebook recently in the past like couple of weeks of people warning not to eat deer meat that looked like it had bubbles in it because it's tuberculosis. And that is a good picture of what the granulomas look like, but I also know zero hunters or zero anybody that would eat meat that looked like it had bubbles on it. I don't know. I'm pretty sure everybody would just assume that it's contaminated and not eat it. So it's a weird thing to have going around. All right, antimicrobial proteins. These enhance your innate defense, and they do this by attacking microorganisms directly or hindering a microorganism's ability to reproduce. The most important antimicrobial proteins are interferons and complement proteins. Complement proteins are my favorite because they're really cool how they work, and they all have like specific names, and they make a pore, and it's cool because they just go in and they make a pore, and then everything goes out of the cell. Interferons are pretty cool too, I guess, somewhat. But interferons are a family of immune modulating proteins. Cells that are infected with viruses can secrete interferons that warn a healthy neighboring cell, hey, I'm infected with a virus. And that can tell its neighboring cell, oh, I need to make DNA or make a protein that's going to block this infection so that I don't also become infected, right? So this is your cell's ways of trying to warn their neighbors that they're not healthy anymore and to block the incoming agent. So interferons will not work on the cell that is infected. The infected cell will produce interferons that enter into the neighboring cell, and in the neighboring cell it stimulates production of proteins that will block the action of the virus so that the virus cannot get into other cells. Okay, so you have different types of interferons. Interferons type alpha and beta will work in this way, but they also activate natural killer cells because natural killer cells are going to kill what kind of cells?
Infecting cells.
What kind of infection?
Iron.
Virally infected cells. So natural killer cells are these cells that you need to activate to kill virus infected cells, and also what kind of cells? Cancer cells. Okay? So these interferons, alpha and beta interferons, are going to activate those natural killer cells to come and kill their neighbor cell that has the virus. And then they're also going to block the virus from reproducing inside of them. You have some other interferons like interferon gamma. Interferon gamma is also called the immune interferon, and it's secreted by lymphocytes, and it has a very widespread immune mobilizing effect, and it will also activate macrophages. The interferons activate natural killer cells and macrophages so that they will indirectly fight cancer. And artificial interferons, because of this possibility, artificial interferons are used to treat disorders like hepatitis C, genital warts, and multiple sclerosis. This is how an interferon works. So if you have a virus that enters a cell, the virus can turn on an interferon gene in that cell. The interferon gene will produce interferon proteins that will go to the neighboring cells. And when it binds with receptors on the surface of its neighbor cell, that is going to cause the neighbor cell to produce antiviral proteins that will block the action of the virus. So as that virus replicates in the first cell, it's not able to get into the next cell. So this is the action of the interferons. The interferons will also go to signal to the natural killer cells to try to kill the virus-infected cells so that it can't just keep producing virus. Okay? So the complement system. So interferons are your cell's ways of communicating with each other to try to block the action of a virus. Your complement system consists of about 20 blood proteins that circulate in the blood in inactive forms. They have numbers C1 to C9 and factors B, D, and P, and then you have some other regulatory proteins. You need to know the complements 1 to 9 because they're going to be important for complement formation that we're going to go over. This provides a major mechanism for destroying foreign substances. Activation will enhance inflammation, and it also directly destroys bacteria. So it enhances both the innate and adaptive defenses. The complement system can be activated by 3 different pathways. You have the classical pathway. So if an antibody will an antibody will first bind to the invading organisms, and then that antibody will bind to the complement components, and that activates them. Double binding is called complement fixation. Once the initial complement proteins are activated, an activation cascade is triggered, and then a pore is formed. The lectin pathway is when lectins are produced by the innate system, and they recognize the foreign invaders. And then when the lectin binds to specific sugars on the foreign invaders, it can start to bind and activate the complement proteins to form that pore. The alternative pathway is when a complement cascade is activated just completely random, completely spontaneous. So when a certain complement factor binds directly to a foreign invader, it can randomly start that cascade to form a pore. Lack of inhibitors on the microorganism's surface will allow the process to proceed. Each pathway will involve activations of proteins in an orderly sequence, and each step catalyzes the next in a cascade fashion. Each pathway converges on the C3 complement protein, which will cleave itself into C3A and C3B. Splitting C3 is going to initiate the common terminal pathway that enhances inflammation, promotes phagocytosis, and causes cell lysis. Cell lysis begins when C3b binds to this target cell, and that triggers insertion of complement proteins that are called your membrane attack complex, or your MAC. And your MAC goes into the cell membrane and forms a hole. So MAC forms and stabilizes a hole in the membrane of the microbe that causes water to flood in and lysis of the microbe. So all of the other ions and all of the important stuff for the cell will start to flow out, and then water flows in, and you are lysing the cell. This is your complement cascade. Remember that all three pathways converge on the C3 complement protein. C3 will then cleave into C3A and C3B. C3B can do one of two things. It can go do opsonization, so it can coat a pathogen surface to act as like a little handle to enhance phagocytosis, or it can cause a complement protein cascade to form your MAC. C3A is going to go to enhance inflammation, and it's going to do that by stimulating histamine release, increasing blood vessel permeability, and attracting phagocytes through chemotaxis. So this is going to be one of your inflammatory signal molecules that signals for chemotaxis. For your MAC protein, you need to know that it's made up of C3b. It starts with C3b, and C3b is going to connect to C5b, C6, C7, C8, and C9. Okay? You need to know these number connections for making your MAC. When C5b binds, it is going to be from the cleaving of the C5 complement protein. And C5a is going to go with C3a, and it's going to do the same thing of enhancing the inflammatory response. The MAC complex is shown down here. It creates a pore. And this pore is going to allow materials to lyse out of the cell and water to flood in. Okay? You usually have many MACs that are going to bind onto a pathogen surface to lyse the cell very, very quickly and get rid of it. You guys are not as loud as I feel you should be, because this is really cool. Complement proteins. And we call it a MAC. Like, it's adorable. There's little proteins and they make a hole, and it's the best thing ever. I love it. All right. Since you guys are so super excited, we'll move on to fever. Should have gotten better wow response from my MAC, I'm just saying. All right, so fever. Fever is an abnormally high body temperature. Everybody knows this. Everybody has had a fever at some point. And it's basically going to be a response, a systemic response, a response for your entire body to an invading microorganism. Leukocytes and macrophages that are exposed to foreign substances will secrete pyrogens. And pyrogens act on the body's thermostat in the hypothalamus. And they do this by telling the hypothalamus to raise the body temperature. You have some benefits to having a moderate fever. If you have a very high fever, it can cause a lot of issues in the body, and it's very bad, right? But if you have a moderate fever, it's going to tell the liver and spleen to sequester iron and zinc, which are needed by those microorganisms, and it's going to sequester them so that it keeps them away from the microorganisms. It's also going to increase your metabolic rate, which will increase the rate of repair for your entire body. The adaptive immune system is a specific defensive system that eliminates almost any pathogen or abnormal cell in the body. Its activities include amplifying the inflammatory response and activating complement proteins. So again, my buddy is on there, my complement is on there, it's important. Some shortcomings to your adaptive immune system is that it has to be primed. So it has to be primed by an initial exposure to a special foreign substance, and priming takes time. This is why your first infection from a virus is going to have a bad reaction of you being sick for a while. But the second time you're exposed to that same virus, you don't get it again because your adaptive immune system is primed and it's going to have a very quick response and kill off anything that's coming in. Okay? So characteristics of the adaptive immunity, it's very specific. It recognizes and targets specific antigens. It is systemic. It's not restricted to the initial site of injury or introduction of the pathogen. It has memory. It is going to mount an even stronger attack to any known antigens. So second and subsequent exposures are going to be very, very strong, very, very fast. You have two main branches of the adaptive system. You have the humoral, which is the antibody-mediated immunity, and you have the cellular, which is the cell-mediated immunity. Which cells do each of these use? It was on one of the first slides that we went over.
B cells and T cells.
Which one uses B cells?
Humoral.
And which one uses T cells?
Cellular.
Okay. Humoral immunity is called humoral immunity because it's going to use the antibodies. Antibodies that are produced by the lymphocytes will circulate freely in the body fluids, and they bind temporarily to target cells, which will temporarily inactivate those target cells, and it marks those cells for destruction by phagocytes or complement proteins. Humoral immunity has extracellular targets. Okay, so this is a very big distinction between humoral immunity and cellular immunity, is that humoral immunity is going to attack things that are found outside of your cells, so whole pathogens that are found outside of your cells. Your cellular immunity is going to focus on your cells and an infection inside of your own cells. Okay? Cellular immunity, you have lymphocytes that act against target cells. They can act directly by killing the infected cell or indirectly by releasing chemicals that will enhance the inflammatory response or activating other lymphocytes or macrophages. Cellular immunity has cellular targets. Your cells are its targets. Your infected cells are its targets. Okay? Big distinction in the beginning. You want to know it because as we go through them and how they work, that is based on whether they're working inside of the cell or outside of the cell. That make sense? All right. So antigens, we start with antigens. Antigens are substances that can mobilize the adaptive defenses and provoke an immune response. Antigens are targets of all of the adaptive immune response. Most of them are large and complex molecules that are not normally found in the body, so they're recognized as non-self. They're usually made up of proteins or portions of proteins that are specific to the cells that you want to kill. So characteristics of antigens, they can be complete or incomplete. If they're complete antigens, we call them complete antigens. If they're incomplete, we call them haptens. They contain antigenic determinants, so something that is specific to that cell that is not found in your body that will signal to your immune system, this needs to go, right? And for the last characteristic of antigens is that they can be a they can be a self-antigen, which means that they're recognized as being a part of that foreign cell. Kind of goes along with the other two. Antigens can be complete or incomplete. Complete antigens have two important functional properties. They have immunogenicity and reactivity. Immunogenicity is their ability to stimulate proliferation of specific lymphocytes. And their reactivity is the ability to react with activated lymphocytes and antibodies released by the immunogenic reactions. So for example, you have foreign proteins, polysaccharides, lipids, and nucleic acids. They're seen on many foreign invaders, like pollen and microorganisms, and your antigens can recognize these. Incomplete antigens or haptens involve molecules that are too small to be seen, so they're not immunogenic by themselves. So small peptides, nucleotides, and some hormones. They can become immunogenic if the hapten attaches to the body's own proteins. A combination of protein and hapten is then going to be seen as foreign. When it's seen as foreign, it causes the immune system to mount an attack that is going to be harmful to the person because it attacks self-proteins as well as hapten. So things like poison ivy, animal dander, detergents, and cosmetics, these can all be like haptens, and they can cause a bad reaction for your body where your body attacks itself, because these are just such small pieces that your body doesn't really go for whole cells to try to attack it. Does that make sense? You have just this small, you have a small peptide or nucleotide and some hormones. All of these are things that are found inside of your body. When they connect to these proteins to form the protein and hapten combination, that's when your body recognizes it as foreign, and it causes the immune system to mount an attack that can be harmful to the person because it attacks the self proteins as well as the hapten. Does that make sense? This is part of your response for allergies, is that you are having a heavy immune reaction to something that you should not be reacting to because it's found readily in your body. Okay? You guys are so good. So we'll stop here for today. Since you guys are half dead. And we'll pick up again on Thursday morning. Yeah. So tomorrow you guys will just have your lecture exam. And then you can go home and drink because you're attacking yourself, because you have to attack your proteins as well as that hapten, right? So that incomplete antigen, when it combines with your proteins, it forms what your body recognizes as a complete antigen, and it will attack anything that it recognizes with that. So that includes some proteins of your own body, and it causes a harmful reaction to the person. So some things that are haptens that cause these bad reactions are things like poison ivy, animal dander, detergents, and some cosmetics, which is why people have regularly have like a dermal reaction to certain types of makeup. So there's usually a lot of different particles in makeups that can cause reactions for people. And detergents are very common. So antigenic determinants are parts of the antigen that the antibodies or lymphocyte receptors bind to. Most are naturally occurring antigens, and they have numerous antigenic determinants that will mobilize several different lymphocyte populations. And they form different kinds of antibodies against them. Okay? So you have large chemically simple molecules like plastics that will have pretty much like no immunogenicity because they're not these complex structures that will that have any kind of like antigenic determinants on them. Okay? So these are your antibodies and antigens. Antibodies look like little Y-shaped proteins that float around and look for a specific antigen. And the antigens have antigenic determinants. The antigenic determinants are going to be like sugars that are found on the surface. And basically what happens is your antibodies, they have two arms, and each arm has a hand that is in a specific shape, right? So they are using like a triangle shape, a circle, and then like a Lego kind of a shape to show you the difference in their structure. So you have this little hand, and it goes around and it looks for something that fits perfectly into the hand. So this is a lot like the active site on a enzyme. So an active site on an enzyme has to have a very specific structure, a very specific shape, and it only fits with like 1 to 3 at the most different types of molecules that it can break apart. It's very, very specific because of its shape. It's the same thing with your antibodies. Your antibodies are each very specific to a certain type of antigen because they have their little hand that is only going to bind to one specific type of antigenic determinant. Okay. Self-antigens, all of your cells are covered with a variety of proteins that are located on the surface, and these are not antigenic to yourself, but they can be antigenic to others if you give like a transfusion or a graft. This is why when they do a organ transplant, what do they put the person on? What kind of medications? What are anti-rejection medications? What do they do?
Prevent the new organ from fighting that person's immune system.
So it's to prevent the person's immune system from fighting the organ. Yeah. So how do these medicines do that? What would they be doing? What action? What would it do to the immune system? So it essentially weakens it. How do you weaken the immune system broadly? Like what are you doing to it? Are you increasing its activity? You're decreasing its activity. So these non-rejection medications, they work by suppressing the immune system, which is also why when somebody has an organ transplant, they basically become like huge germaphobes. Their immune system is so suppressed that a common cold virus can kill them because their body will not be able to mount the correct immune response to something as simple as a cold virus. So your body is usually very, very good at handling things that you normally get sick with, like a cold virus, and you'll only be sick for like a week at the most kind of a thing, right? But for people who have had an organ transplant and their immune system is suppressed to a level that they will not attack and reject that organ, then they have like something as small as a cold that will just kill them. So when people get an organ transplant and they're super germophobic, don't be offended if they don't shake your hand. They're just trying to protect themselves. Okay? All right, so the self-antigens are very important so that you don't have an autoimmune response, so that your body does not attack itself. When people have a mismatch between their antibodies and their self-antigens is basically the science behind why people have autoimmune disorders. Their body does not recognize self and attacks itself. Okay? So there's one set of important self-proteins, and these are a group of glycoproteins, and we call these the MHC proteins. The MHC proteins are the major histocompatibility complex. They're unique to each individual, and you have two main types. Each main type is going to go for one of the types of adaptive immunity. Yeah?
Is that why, like when some people get organ transplant, after a while it starts rejecting? Is it because your body's not responding to it?
It's because their immune system is attacking the organ. So usually when we do organ transplants, you are looking at trying to basically make the organ as similar to the person's body as possible. So when they give an organ transplant, they do like blood typing and try to match the individuals as close as possible, right? So the closer that they are matched, the less you're going to have a rejection. But even when you match it very, very closely, you're still taking proteins that have somebody else's self-antigens and putting it into another body. So when the organ gets rejected, it's because they're too different, and their body, like even suppressed, is not going to ignore the organ and just attacks it to the point that function is lost. Okay? So then you have organ rejection.
Sorry to interrupt. Amanda's door's locked. I bring
Oh.
this for her.
Okay.
Okay,
Thank you. All right, so for the major histocompatibility complexes, these are unique to each individual, and they have grooves that will hold a piece of self-antigen or foreign antigen. The T lymphocytes can recognize only antigens that are presented on the MHC proteins. So your T lymphocytes rely on these MHC proteins being there. The B cells are not going to rely on it as much, but the B cells can also react to certain MHC proteins. All right, so adaptive immune system will involve three crucial types of cells. You have the two types of lymphocytes, B and T, and then you have the antigen presenting cells. Your antigen presenting cells are your APCs. You need to memorize this now. APCs, antigen presenting cells. They're going to come up a lot as we go through how these cells function. Antigen presenting cells do not respond to specific antigens. What they do is they find something foreign, they break it up, and they take its antigens and they present them on their surface. So you have these antigen presenting cells that are just going around to all of your other cells and going, hey, I found something. Hey, take a look at this. I found something. And then your antibodies will come and like check it out and like see like, oh, does that match? I don't know what that is. Goodbye. And then another one comes in and checks, and I don't know what that is, and goodbye, right? So your antigen presenting cells are just certain types of cells that will take an antigen and present it on the cell surface for your immune system to come and find it, okay? They play an essential auxiliary role in immunity. They are very, very much needed to mount a response. All right, so for lymphocyte development, maturation, and activation, we're going to go over this fairly briefly, okay? So T and B lymphocytes will share a common development, and they have common steps in their life cycles, but they're just very slightly different because you have some that are going to become T cells and some that become B cells. There has to be some kind of a small distinction between the two. So for their 5 general steps, the origin is going to be both lymphocytes will originate in the red bone marrow. Okay, this is something that you guys should remember from the blood lecture. Your all of your blood cells are going to originate in the red bone marrow, but your B cells and T cells are going to mature in different areas. Okay, so the origin for both is in the red bone marrow. Where they mature is going to be different. Lymphocytes are going to be educated. They have to be primed. They have to be taught what self is so that they don't attack your body. And so they have to go do this process and learn what is self and what is something to protect. And they're going to learn this in 2 different areas. So they're going to mature in the primary lymphoid organs, which are going to be the bone marrow and the thymus. So that's another important thing. And I think when you guys watch Lecture 20, I say that multiple times, that your 2 primary lymphoid organs are your bone marrow and your thymus. So these are very important. This is your red bone marrow and your thymus. Immunocompetence is when the lymphocytes can recognize only one specific antigen. So B or T cells will display only one unique type of antigen receptor on the surface when they're mature. And so they can only bind to one specific antigen. During maturation, they're also going to be taught self-tolerance. lymphocytes have to be unresponsive to your body's own antigens. If they respond to your body's own antigens, they are destroyed and they are not allowed to reach maturity. Okay? Or they shouldn't. If they do, you start having autoimmune disorders. The T cells are what are going to mature in the thymus, and they do this under negative and positive selection pressures, or basically negative and positive tests to make sure that your T lymphocytes are recognizing self cells, because your T cells have to go through a heavier process, because what is their target? What do they work on? The cells of your body. If you have something that is going to be attacking the cells of your body, and you want it to only attack the infected cells of your body, your T lymphocytes have to go through a more rigorous testing system for priming to make sure that they are not going to attack your healthy cells, and they need to just attack the infected cells. Okay? So the positive selection process is going to select the T cells that are capable of recognizing the self-MHC proteins. So this is called MHC restriction. And if they can't recognize the self-MHC proteins, they get destroyed by apoptosis. The negative selection is basically going to do the same thing. It's just like a secondary process and looking for a different thing. Or looking for the opposite. So this prompts apoptosis of T cells that will bind to self-antigens that are displayed by self-MHC. So first, it's can you recognize this as self? If you can't recognize this as self, you're going to get killed. And then if it does recognize it as self, then it's going to give it self again and be like, do you recognize this as bad, or is this self? And if they recognize it as bad, they get destroyed. Okay? So it's like a double system to try to make sure that your T cells will recognize your self cells and not accidentally destroy the cells of your body. So the negative selection is called clonal deletion, and it ensures that you have self-tolerance, that you are not going to attack your own cells. So this shows each process. You have your positive selection, where the T cells have to be able to recognize the self-major histocompatibility proteins, so they have to be able to recognize self-MHC. If they fail to recognize the self-MHC, they are going to be signaled to undergo apoptosis, or death by cell suicide. If they recognize the self-MHC, then this results in survival, and the survivors are going to proceed to the negative selection. So positive selection is first. Do you recognize yourself? Yes, go on. So then they go into negative selection. In negative selection, they must not recognize self-antigens as bad. Okay? So if they recognize the self-antigen as bad, then they are going to undergo apoptosis. If they fail to recognize the self-antigen as bad, then they will go on for survival and continue through maturation. Okay? So you can see from each of these, for recognizing self, if there's no binding here, then it doesn't recognize a self-antigen, right? The binding here is not complete, so it's going to recognize that this is self-antigen, the proteins match up, but this doesn't match up, meaning that the antigen itself is not bad. Does that make sense? And then when it goes through negative, if that matches up, then the T cell is recognizing it as bad and is going to try to kill a self cell, which you don't want to have happen. But if you have, the cells are still going to recognize each other, but it doesn't recognize it as bad, they go on to mature. Okay? Does this make sense for positive and negative selection? All right, so the B cells are going to mature in the red bone marrow. Not as much is known about the process of B cell maturation. Only immunocompetent B cells are allowed to mature, and those that are self-reactive are eliminated by apoptosis. For this one, we just call it clonal deletion because we don't really know a whole lot about it, and so it's just clonal deletion. Okay, so you have two of the steps for lymphocyte development. You have the origin, and then you have maturation for both of them. And then the third step is seeding secondary lymphoid organs and the circulation. So the matured B cells and T cells, the immunocompetent B and T cells, They're not yet exposed to an antigen, so we call them naive B cells and naive T cells. So your naive B cells and your naive T cells have to be exported from the primary lymphoid organs. And they are going to seed in secondary lymphoid organs where they wait to come in contact with an antigen and learn it. Okay? So they're going to go seed in things like the lymph nodes and the spleen. And this increases their chance of encountering an antigen and learning it. The fourth one is antigen encounter and activation. So the naive lymphocytes, when they first encounter an antigen, it triggers the lymphocyte to develop further. The lymphocyte is going to be selected to differentiate into active cells that bind to that specific antigen. And this is clonal selection. Okay? If the correct signals are present, the lymphocyte will complete its differentiation into an active cell. Make sense? Everybody's okay so far? All right, so then you have proliferation and differentiation. So once a lymphocyte is selected and activated, it proliferates. It's going to form an army of exact copies of itself. It's just going to have this, it meets up, it touches, and you have an explosion. It just proliferates like crazy. You guys can watch videos on the immune system. There's a lot of really good animations on YouTube that will take you through this and show you, and it just like goes around, and then the second that an antigen gets there and it touches with the right lymphocyte, that lymphocyte will just explode into proliferation. So it makes an army of clones, and those clones will become effector cells that will fight the infection. So they will clone like crazy, and then they differentiate. And as they differentiate into the fighters that are going to go kill off the infection. A few of them are going to become memory cells, and the memory cells are going to be there and primed and ready for if a second attack comes, they can activate immediately and attack the invasion. Okay? So B and T memory cells and effector T cells will circulate the body continuously. Right, let's see, where are we? 5, let's go like 5 more slides. All right, so antigen receptor diversity. Genes, not antigens, will determine which foreign substance the immune system will recognize. There's a variety of immune cell receptors that are a result of acquired genetic knowledge of microbes. There are about 25,000 different genes that code for up to 1 billion different types of lymphocyte antigen receptors. So you have a huge adaptability to recognizing horrible, or not horrible, but foreign antigens so that your body can attack it. So that huge variety of receptors, the gene segments can be shuffled around, and this results in many combinations, almost an unlimited amount of combinations, because there's just so many combinations you can have. You can fight off pretty much anything. So this is an overview of B and T lymphocytes. Again, the tables are helpful for comparing the two and making sure that you know the differences between them. All right, so your antigen-presenting cells. These will engulf an antigen, and then they present fragments of the antigen to the T cells for recognition. So there are some major types of antigen-presenting cells. You have dendritic cells, you have macrophages, and you have B cells. Dendritic cells are found in your connective tissues and in the epidermis. Macrophages are widely distributed in the connective tissue and the lymphoid organs. And B cells will present antigens to helper T cells to assist their own activation. So B cells are also also going to act as antigen-presenting cells. So when a B cell encounters a target antigen, it provokes your humoral immune response. Antibodies that are specific for that particular antigen are then going to be produced. B cells are activated when the antigens bind to the surface receptors, and this cross-links them, and it triggers receptor-mediated endocytosis of the cross-linked antigen receptor complexes, and this will start clonal selection, and that leads to proliferation and differentiation of the B cell into the effector cells. Most clone cells will become plasma cells. Plasma cells are antibody-secreting effector cells. Okay? So your B cells become your plasma cells. Your B cells will become your plasma cells. Okay? Plasma cells will secrete specific antibodies at a rate of about 2,000 molecules per second for 4 to 5 days, and then they die. Antibodies will circulate in the blood or the lymph, and they bind to free antigens, and that marks them for destruction by your phagocytes, and in particular in your innate immune system, and it will also trigger other adaptive mechanisms like your complement proteins. Okay, so this is your primary, or your responses for clonal selection of a B cell. The antigen will bind to only one type of B cell. It has to be specific to the receptors that are on the B cell. And then that will proliferate to form clones. And those clones will proliferate into memory B cells and plasma cells. The plasma cells are only going to hang around for about 4 days. And their primary job is just to produce antibodies in huge amounts. Okay? And then the plasma cells will die. The clone cells that don't become the plasma cells, those become your memory cells. They provide immunological memory so that they can mount an immediate response to any secondary or future exposures to the foreign substance. So your memory B cells are what circulate and look for the antigen again, and if they come in contact with the antigen again, it mounts an even bigger response where the proliferation is even faster. All right, so we'll pause here for picking up again on Monday. Okay. So you guys have, I need to get your dissection out. The dissection is going to be up here in the front.